Oh, I barely have my voice. So today was a really busy day. I had a whole bunch of stuff going on, and I just got back from freaking the bar. Like I went to a Taylor Swift night, and I thought it was gonna be kind of like fun, cute, cool. It wasn't fun, cute, or cool. It was like really crowded, and everybody was pushing up on me, and everybody was sweating, and everybody screaming, and I didn't feel very nice. But I went with my friend, and we had fun. Because we did, like, an event before that, and it was actually pretty nice, and, I was so traumatized from Taylor Swift that I went home and I blasted EDM from my speaker for, like, 30 minutes, and now I'm here ready to study because I just wrote down a whole agenda on my whiteboard, and I figured out that I have way too much to do on Friday and to do on Friday. Like, I have a lot of things to do before Friday and by Friday that I can't just keep slacking like this. So this lecture is going to be about the biolab 2 and 3. I already finished it. I already did it this Tuesday, but I just finished the notes. Well, I finished the notes before I left the house. I wasn't gonna leave the house without finishing my freaking 4 pages of notes on my lab. And honestly, I don't even know if this is like required or necessary, but I'm gonna do it anyway because I don't know, academic weapon, I guess. Here, I don't know if I'm the one crushing the academics or the academics are crushing me, but we're just going to dive into it. This was 33 pages of the document and 43 slides that I went through to write these 4 pages of notes, and it was a headache, and I didn't want to do it, but I did it anyways. So we're just gonna get into it, right? Okay. And yeah, I'm miserable. Like I was sick and I have so many things to do. I might just pull an all-nighter tonight to catch up on all the chemistry stuff that I've been missing out on. See, my face is greasy as frick, and my body hurts. Oh, and I'm hungry. I don't have any food, and it's like freaking 11 p.m. So that's great. College things. Okay, let's get into it. I am not going to delay myself further than this. Let me pull up. And the going to the bar just really whooped my ass because I'm like tired as frick. Okay, and excuse my room. It's not gonna look good because it's freaking at night, and I'm not going to. I'm just not gonna look at it. Okay, so the lab 2 and 3. It's about non-vascular and seedless vascular plants. So we some terms and reminders you gotta know about these plants is they go through sporic meiosis. They don't go through gametic and go through the other type. They go through sporic meiosis, which is also referred to as alternation of generations. I wrote alteration on the other lecture. Don't listen to me. It's alternation of generations, right? Okay. So why is it called alternation of generations? Because it alternates between the gametophyte, which is the haploid and gamete forming type of cell, and then the sporophyte, which is the diploid and spore forming type of cell. So gametophyte is haploid, and then sporophyte is diploid. So gametophytes become gametes, and sporophytes become spores, I believe. And then yeah, okay. So we're first gonna go through the non-vascular plants. There's 3 phyla of these. We got Bryophyta, Hepatophyta, and Anthocerophyta. And these are also known, all of these are known as bryophytes, not to be confused with the Bryophyta phyla. That's the phyla, but all of these are known as bryophyte. I just got a text, so give me a second. Okay, and yeah, we're gonna talk about the growth of a bryophyte gamete or gametophyte. I'm sorry, I hate these words. They're just so long. Okay, so first, the haploid spore, which is the myospore, is gonna be germinated, right? And I don't know what that means, but we're just gonna know that there's germination of a haploid spore, which is called a myospore, and then there's a formation of a protonema. The plural form is protonemata, protonemata, whatever. But it's a multicellular filament on the cell. And thirdly, we have mature gametophytes are autotrophic plants with a simple plant body. Okay, so this is not just a step, it's just like a little note that's saying like the mature gametophyte, when it's like done developing, they are autotrophic and they have a simple plant body, which is a thallus, and then it doesn't have true roots, stems, or leaves. Another note, gametes, which are produced by mitosis, are protected from desiccation by a layer called gametangium. So the plural is gametangia. And it's produced on the main axis of the side branches of the mature gametophyte shoots. So yeah, the gametes are protected from like the scratches, I guess. Anyways, sperm are produced in the antheridium, which is the male gametangium, and a single egg is produced in the archegonium, so the female gametangium. So basically, the antheridium is the male, and then the archegonium is the female. I remember anthers, kind of like some like antlers, and then comes male E, and the archegonium. I think begonia means like egg or something, and that's supposed to be very female like. All right. So some plants are really like they're really bisexual, like they swing both ways, this basically means that the male female gametangium are produced on the same gametophyte. So yeah, the same like gamete cell is going to have both the male and female gametangium, right? But unisexual is this, but the male and female gametangium are produced on separate gametophytes, so they're unisexual. So I mean, it might be hard to remember, but basically, bisexual is like it has two on the same gametophyte, and unisexuals each gametophyte has a different, gender of gametangia. Okay. The next step, the fertilization. So the zygote develops into an embryonic sporophyte, and then the meiosis in the sporangia results in myospores that are dispersed. So that's a lot to take in, and I'm just not going to explain it because I don't have time. I don't want to get into it yet. But I'm just gonna read it out, and then we'll go over it in the end when we need to deal with it later. Okay. We're gonna go into the first phylum. So we got Bryophyta. So these are true mosses, and basically it looks like a little grass, like a little plant on the grass. So let me see if I can draw it for you. Uh-oh, I dropped my entire note thing. That's not very sexy. Oh, my paper's on the floor. Okay, I got it. All right, so I'm gonna draw you a bryophyta type of plant. Yeah, I am hungry and sleepy. Okay, so very cute, very simple. We're just gonna keep it simple, just like that. This is ugly. Can't draw for anything, but this is what it looks like, right? Okay, so this is gonna be the foot, right? This is the foot. And then this is going to be called the seta or the stalk. And then this is the capsule or the sporangium. This entire thing is the capsule, right? And this entire thing right here, this is the sporophyte. Sporangium, sporophyte, right? And give me a second. Give me a second. Okay, yeah. So that's the sporangium. This is sporophyte. And this little grass-looking thing that's beneath the foot of the spor uh beneath the foot of a sporophyte. This is a gametophyte. This is gametophyte. This is sporophyte. This is the foot of the sporophyte. This is the stalk or the seta of the sporophyte. This is the capsule or the sporangium. Okay. Okay. That's all you need to know for now. Uh, look at that. I hate when they do this. Okay. So the capsule is covered by a cap-like structure, which is called the calyptra, which is the remnant of the archegonial wall, which is basically like so it means it's like a cometophyte tissue. And the operculum is the capsule lid, so I'm assuming that's like the very top of it. And the peristome is the tooth-like projection on the inner opening of the capsule. And I believe this is supposed to spread like the spores around and stuff. All right. So that's what it looks like, and that's what a bryophyta looks like. Okay, so now we're gonna move on to the phylum Hepatophyta, which is like also known as liverworts. Of course, since you're in medical, you know that hepat means liver. So yeah, I don't know why they're called liverworts, but basically it looks like a lettuce with like cups on it, like suction cups, pretty much like little like toilet suction cups. I don't like it. It's pretty cool. I don't like it. So for the Hepatophyta, the reproduction occurs by fragmentation of the thallus, which is like the stalk, I believe, and by the gemma, which is small outgrowths that can develop into entire new plants. So the gemma are like the cups on the plants. And we're gonna talk about antheridiyophores and archegoniophores. Of course, you've heard this before with, I believe, antheridiospores. Am I lying? I am lying. Antheridium and archegonium. So now instead of that, we're talking about antheridiyophores and archegoniophores. Same deal. Antheridiyophores are the upward extensions of the male midrib, and they're disc-shaped. And the archegoniophores are the upward extensions of female midrib of phallus, and these are umbrella-like tops. It's about elaters or elaters, I don't know how to pronounce it, but they're non-reproductive cells that are formed in the sporangium along with the spore mother cells, and they help aid in the disposal of spores. Okay, last phylum of the non-vascular plants. So this is the phylum Anthocerophyta. These are hornworts, and they can photosynthesize, and I don't really know what they look like, so you have to search it out. I believe they're no, I don't believe anything. So yeah, just know that. Okay, we're gonna move on to the seedless vascular plants. Man, I'm getting so tired. I don't want to talk anymore. Like this is the best way I can study, and it's like the most efficient way I can study, but I don't even have any motivation right now. I feel sad. I feel tired. I want to sleep for once. Maybe I should sleep for like 4 hours. Maybe I should just sleep until I wake up again, and then I can just study. Well, it's a lot of studying I gotta do because, like, maybe I will finish these notes, write some anthropology notes, like try to finish anthropology while listening to music or like watching something, and then tomorrow I can just focus on chemistry, hopefully, for the whole day and not get distracted. That sounds very ambitious. Anyways, we're talking about seedless vascular plants. There's four of them. So these are distinguished from bryophytes by a possession of an efficient water and food conduction system. So yeah, they have a food and water conduction system, and the bryophytes don't. So they also have vascular tissues. So they have xylem for water and minerals, and the flow is from the roots upwards only. And they have the phloem for carbohydrates and other nutrients, and these flow in whatever direction that like the plant needs, like up or down or whatever. So just kind of think about like for water and minerals, they only go up because they need to go up. Like water comes from below up, and then for like nutrients and whatever, like you can just go anywhere pretty much. I like to kind of think of it as like when you drink water, most of it just kind of goes down, you know. But when you get nutrients, like you gotta get nutrients up to your brain, you gotta get food up to your brain, stuff like that. Not entirely accurate, but that's how I remember it. So we're gonna talk about the first phylum, which is Lycophyta. These are called club mosses, and they have strobilus, which is strobili, and they're kind of like cones, you know, like think pine cone. So these are cone-like structures that consist of a cluster of sporophylls, which are leaves that bear sporangia or sporangiophores. That is a lot of words that I will not get into, but just kind of think of it as a pine cone, kind of. So that's the club mosses. And we'll talk about the Psilophyta. So these are whisk ferns, and they're the most primitive vegetable plants, so they don't really have leaves. But they have dichotomous, which is two-way branching leaves, and they also have sporangia or sporangium, which where spores are produced in. And they have rhizoids too, which are root-like structures and produced on rhizomes, which are horizontal underground stems. So rhizomes produce rhizoids, sporangia produce spores. All right, we're gonna move on to the phylum Anthophyta. Anthophyta, horsetails. They're horsetails. There's only one genus with 15 species, and they have nodes, which is an attachment site of a stem and a leaf. So you know, like there's a stem, right, and then there's a leaf. Here, I have an actual plant to show you. It's like pretty cool. I don't know. I'm a biology major, so I have plants obviously everywhere. Anyways, so there's a stem, right? There's a leaf. This is the node right here, the attachment site of the plant. My flowers are dying. It's like a cemetery over here. Man, look at me. I am oily. I'm oily London. I'm glittery and I'm oily. It's like I'm blinding you. Oh my gosh, I'm blinding myself. Blinding myself with all these notes that I wrote. Oh my gosh, look at it. This is pain. This is pain all in my brain. I did not expect this from college. It's fun. I like it. I get to do a lot of things that I didn't get to do when I was in college. I have a whole bunch more experience and responsibilities and freedom and friends and plans and a lot of more events and opportunities for myself. But I also have a lot of suffering, and that's what I have to deal with every single week for the rest of my 8 years here. And I'm not really doing too well at 11 p.m. Let's continue now. So for the phylum Anthophyta, which are horsetails, I already talked about the nodes, but there's also internodes, which is basically the area between the nodes. Okay, look, with that, we have finished with 2 pages of notes. Congratulations. 2 pages, 20 minutes, pretty good. At this rate, we might be finished by 40 minutes. And then I could go to sleep. Or I can like write notes and then go sleep. Or I could just ball the whole night and wake up and wake up late. I think I'm just gonna do like option 1, like go to sleep and then no, option 2 and write my notes and then wake up. Um, when I want to wake up because I sleep early and then I can finish my chemistry notes because I'm literally losing my mind. Yeah. once we watch this back when I'm actually like human and sane again, and I'm just not gonna be functional at all. I'm just gonna be like dead. Anyways, so we're gonna move on to the phylum Pterophyta. So these are ferns, and this is the largest group of the vascular plants, right? And they have rhizomes, which is underground stem. Again, I already told you about the underground stems. And the fronds, they have fronds, which are large leaves on them. So yeah, think about ferns. They have large leaves, right? Those are called fronds. Okay. But each frond is a megaphyll meaning big leaf, consisting of a leaf blade and a petiole, which is a leaf stalk. So you know what a fern looks like. So there's the petiole, right? And then they have like they have like leaf blades, you know? Yeah. Anyways, the leaves are simple or compound, and ferns are homosporous. They can either be homosporous or heterosporous. And the spores, again, are born in sporangia. The clusters of the spores in the sporangia are called sori, or singular is sorus, like Brachiosaurus, but it's spelled S-O-R-U-S, which is kind of boring. Anyways, the fern life cycle begins with meiosis and the formation of a meiospore, which is haploid. And it germinates into a filament of cells, which is the protonema. And then the filament undergoes cell division and develops into a flat thalloid structure, which is also called a prothallus. And then it ultimately becomes a mature gametophyte. Okay, so some terms to go over. We have homospory versus heterospory. So homospory produces a sporophyte that produces only one type of gamete, and heterospory is the sporophyte produces two two different ones. Okay, and then the megaspores versus microspores. The megaspores are female gametophytes, and the microspores are the male. We got megaphyll and microphyll. So mega is big leaves, microphyll is small and simple leaves. Okay, cool. All right, we'll talk about beans. Okay, I hate beans. I'm allergic to all of them, but we are going to learn about them anyway. So I'm gonna draw this, and I'm gonna draw this. And if anybody comes to me about how my drawing is nasty, I will have to kindly ask you to shut up because it is not my time of night right now. Okay? Okay. Alright. So this is the bean seed. Okay. Also called a dicot. Pretty cool. So this epicotyle right here. I'm gonna draw an E. Oh man, this is not working. It's okay. But this here, let me just draw a star. That just made it worse. Anyway, you know right here, this small leaf right here is the epicotyle, also known as a plumule. And this is a young stem or the leaf tips. So like if this is baby leaf. Okay, well don't do that. Okay. And then we have the hypocotyl, which is the axis part below the otolodon attachment. I don't know what that means, but it's just right here. Like, I think it's just right here. Yeah, it's just kind of like where the thing is attached. Oh, cotyledon. I don't know why I wrote. Oh. Anyways, the cotyledon is the embryonic leaf, so this is the cotyledon. The big leaf right here. So the hypocotyl is the area below the attachment site. So like right here. Anyways, radicle, we have the radicle, which is the embryonic root tip. It becomes the root. It eventually becomes root. So that's right here, the radicle. And then we have the seed coat, which is right here. And it's the protective coat of the embryo. Very nice. Now we got a different group to move on to. We got gymnosperms, which are also called naked seeds. Got four of these. And first phylum we'll talk about is the Psychophyta, right? Okay. And one thing to know about Psychophyta is they're dioecious, which means male and female structures are produced on separate individuals, right? And it should have a little tie to heterospory, maybe? No? I mean, this one's just male and female structures. Oh, I think it means bisexual, unisexual. Anyways, so we have pollen and ovulate cones. So ovulate cones are much larger than pollen cones because pollen cones are small and men are smaller than women. And then I have the word megasporophylls, and I wrote WTF next to it because there was no description, no context, no information on that, so I just wrote WTF. And I guess we just gotta know about megasporophylls. Uh-huh. All right. The next phylum we're gonna talk about is Ginkgophyta, which is the only species. Oh, there's only one species of these. There's Ginkgo biloba. There's only one living species, which is Ginkgo biloba. So it's deciduous, means it sheds leaves annually, and it's dioecious. So yeah, so separate male and female sporophytes, and they have small microsporangiate strobili, which is like small male cones, and then large female seeds. Next phylum we got phylum Gnetophyta. These are unusual and bizarre plants, but one example of this is ephedra, which is the original source of ephedrine, the drug. Very nice. Next phylum we got. Oh, it's the last phylum. Is this the last one? Oh my gosh, it's the last phylum. All my notes. Goodness gracious. We might finish this in 35 minutes. It's okay. Let's not get too hopeful and keep talking and not get distracted. So we got Coniferophyta. Yes, these are conifers, and pines are one of them. Oh, they are, not aren't, but they are one of them. And they have cones or strobili. So by now you should know that strobili means cones, or strobilus is a cone. So we're gonna talk about some seed development in pines, right? This is gonna get a little bit complicated. There was like 2 pages of explaining the process. I didn't understand it, but I tried my best to kind of like word it in the way that I would understand it in my notes. And here I am trying to explain it once again to myself. It's not as confusing as grammatic meiosis and all this stuff. Okay. So the micro and megasporangia are produced in separate strobili. We already said that. And the male strobili are made of microsporophylls, and they're also known as pollen cones, staminate cones. If you want to sound like a genius PhD, you say microsporangiate strobili, but basically it just means male cone, you know. But you know that they're made of microsporophylls, and the microspores are produced in microsporangia, and they develop into a pollen grain, which is the microgametophyte. So we got strobili, they're made of microsporophylls. Okay, I'm gonna sound like a PhD for a second. Right. So in essence, the microsporangiate strobili, they are made of microsporophylls, and the microspores are produced in the microsporangia. These develop into pollen grains, which is the microgametophyte of the male strobili. Doesn't that sound genius? But do I know what the heck I'm talking about? Is the question. I don't think so. So we're gonna try to explain this in better terms. So the male like cone is made of like, see, I can't even explain. So they're like this kind of like microspore things, I think. And then yeah, they're made of microspores. So the male strobili are made from microspores, and the microspores are made in the microsporangia. So if you hear angia, I think it just means like the spores are made in there, and then it develops into the pollen grain. So like it's the gamete basically, but it's like a small gamete, so it's a microgametophyte. So same deal, but with females, it's a little bit more complicated. So I just explained to you the male strobili. Now we're talking about the female strobili. These are also they are made of megasporophylls, not microsporophylls, and they're also called seed or ovulate cones. So for the males, it's called pollen or staminate cones, and females, it's the seed or ovulate cones or megasporangiate strobili. Yeah, exactly. Get that. You couldn't be smarter than me. Not at 11:29 and 17 p.m. on Wednesday, September 6th, after attending a meeting with a professor and his class, and eating dinner, and going to a museum, and going to a bar, and almost like dying of hunger, and dying of hunger right now. So it's going great. Okay, let's continue. Okay, so the megasporangia are covered by integuments, which are tissue layers. It's pretty cool. We're protected. And the megasporangium are it's also called the nucleus, not nucleus, it's nucleus. Nucellus. Nucellus. I couldn't care less. Anyways, the entire structure is an ovule. hmm The entire structure is an ovule, and then the nucleus is the megasporangium, which produces the megasporophylls, I believe, which takes part in being part of the sporangiate strobili. Okay, moving on. So what makes these seed plants unique from the plants that we studied before is the microspore and the seed plants are they're not discharged. So I think like the seeds aren't discharged. They're retained in the megasporangium, and then it gives rise to the mature megagametophyte, and like also has the archegonia, becomes a sporophyte embryo after fertilization. So it just all kind of happens right there. There's no like movement, pretty much. I think is what I was trying to say. A little note, last thing, it's the last thing we're talking about. A little note is the pollen grain is extremely reduced male gametophyte. So it contains two haploid cells. There's a tube cell and the generative cell. The generative cell eventually will give rise to two male gametes, which would be the sperm. And with that, we are finished with our notes. I'm going to wash my face and try to finish my anthropology notes. No, I haven't even started. So I'm going to try to get those done. And then once I get those done, maybe I can relax a little bit, go sleep, eat. No, I'm not eating. It's too late. But yeah, um, if you watch this, sorry, I'm not doing too well. But tomorrow I might. And definitely this Friday is gonna be a lot of fun. This Saturday is gonna be a lot of fun. We're gonna have a lot of fun soon. So yeah, bye.